All right. Thank you for your for being here and for the amazing primers that everyone here in this session have. I'm going to finish this session with talking about modeling immune endometrial crosstalk in MPS systems. And I would like to thank the organizers and for an amazing conference thus far. As we've previously been primed to, the human endometrium, which is responsible for reproduction, is really a complex, hormonally responsive, and hyper-regenerative tissue. Now, the endometrium is this layer which actually, over the course of 28 days, scarlessly regenerates over 1 centimeter, and it rests upon the muscle layer, which is called the myometrium, which is responsible for contraction and for birth. And it's comprised of primarily a luminal glandular epithelium with a bunch of stromal cells, and there's tissue-resident macrophages, NK cells. And these kinds of interactions are of particular interest because they change over the course of the 28-day menstrual cycle. In particular, the stromal cells undergo the decidual process, which prime the tissue to be prepared for the pre-implantation embryo. Now, what happens when these processes are dysregulated? Well, one of a fairly prevalent, but actually publicly not really recognized disease is called endometriosis. And the pathology of this disease is it becomes lesions on ectopic lesions within the peritoneal cavity. And it's thought that the process of the menstrual shedding is dysregulated in this disease, such that when there is reflux menstrual tissue that leaks into the cavity, it attaches and forms these lesions. And what's interesting is that 90% of women experience some form of reflux menstrual tissue. However, only 10% globally, well, that's a fairly large percentage, actually develop and maintain these lesions, right? And so the implication is that there is immune clearance deficiency within this disease, right? And it presents itself as these really painful, so there's dysmenorrhea, and also contributes to infertility. And as I said, it's also hormonally implicated. And so how in the Griffith lab and in the Laufenberger lab, we're very concerned or interested in properly modeling this disease. So how do we actually model this lesion microenvironment? Especially how do we move into MPS or physiologically relevant systems? Well, if we consider what I was just talking about, maybe we can provide some glandular epithelial organoids, maybe seed stromal cells, and then have these monocytes and macrophages. It quickly becomes a pretty complex system. So when I entered the lab just shy of a year ago, I was particularly interested in kind of stromal monocyte crosstalk because of what was previously identified as this kind of immune inflammation component, which is dysregulated in disease. So I'm going to go over this this entire disease modeling pipeline with you today. So first, we'll start with the hypothesis generation and then go through some of the very key tailored ECM environments that we generate in the lab and how we actually combine these with patient-derived cells, both stromal cells, epithelial cells, and monocytes derived from patients. And then first, place them into static three-dimensional culture. But ultimately, what is a developing story is integrating these systems into MPS devices. And there's just like a feedback loop that ideally through understanding more of these mechanistic drivers behind disease. So let's first begin with the hypothesis generation. Very recently, Lauren Baugh, which is a former postdoc in the lab, did a multi-omic screen of uterine biopsies, both from patients without endometriosis and with endometriosis. So she did single cell RNA sequencing and multiplexed proteomics, and then utilized pathway analysis and also NicheNet to understand ligand receptor interactions. And the upshot of all this is that she found that TYRO3, which is a gene which is implicated within immune clearance response, is significantly downregulated in disease, right? And so this, so this kind of pointed to a potential immune deficiency in terms of tissue clearance, which I previously described is phenotypically present, but actually molecularly hadn't been characterized up till this point. Similarly, others in the lab, in particular Juan Neco, who just recently got a faculty position at Tufts University, utilized a model that I'm going to describe a little later, where he stimulated co-cultures of stromal cells and epithelial cells with an inflammatory stimulant, IL-1 beta, and induced a diseased phenotype well known within endometriosis, which is progesterone resistance, right? And so this leads to the thought that, okay, it replicates this disease characteristic, but the question when I came to the lab was very curious was, how do stromal monocytes interact, right? Because in a pro-inflammatory environment, monocytes are known to potentially secrete IL-1 beta. So can we gain insight into the mechanisms behind endometriotic disease by modeling these interactions in vitro? So this was the hypothesis, that diseased stromal cells disrupt immune clearance mechanisms by dysregulating monocyte activity in a cycle-dependent fashion. So now I'll quickly talk about how we model these systems, first with the PEG-based ECM. So if we are to look at tissue compartment crosstalk, you need a low noise environment. And typically in a lot of animal-generated or animal-derived ECMs, it's difficult to actually know or control lot-to-lot variability. So we in the Griffith lab utilize a PEG-based hydrogel, which is based on an 8-arm reactive PEG macromer functionalized with a vinyl sulfone group, where we can add binders such as SYNK-RGD or G-Foger, which mimic collagen and fibronectin, and fibronectin binders or basement membrane binders that replicate collagen-4, laminin, and fibronectin, and generate these hydrogels, which are very reproducible and which are also MMP cleavable. And we can tailor not only the constituents of this gel, but also the stiffness of the gel, and tailor it to the environment that we are trying to engineer. So this gel has been utilized in a lot of not only cold cultures, but monocultures. So Alex Brown published a paper demonstrating endothelialization and vascularization, and Alan Kahn had a beautiful talk back on Wednesday that you can look at back in a recording. And Juan Neco recently submitted to bioarchives both the stromal and epithelial cold cultures. And today I'll talk a little bit more about the monocyte stromals. But why do we even move into 3-dimensional gels? So quickly, I'll talk about that. So if we were to look at the decidual response of stromal cells in 2D versus 3D, we actually see that morphologically in 2D, they become very flat. In 3D, they're a little bit more biomimetic, but and a characterization of that is that the secretion of prolactin is significantly increased in three dimensions versus 2D culture, right? And this is work done by a talented undergrad called Kira Butley at MIT. So now I'll talk about how we combine the stromal cells and monocytes, and what kind of insights that we gain into endometriotic disease. So in our co-culture model, we derive the stromal cells from endometrial biopsies from our collaborators at Newton Wellesley Hospital, and we gather CD14 positive monocytes from our collaborators at MGH, and we combine them. So we pre-decidualize some of these cells, and we co-encapsulate them to replicate the 3 phases of the menstrual cycle. So the non-differentiated cells would be co-encapsulated with monocytes in this proliferative phase model. And then in the secretory phase, we pre-decidualize these stromal cells, co-encapsulate. And then in the Menzies model, we have the pre-decidualized stromal cells and then withdraw the addition of MPA, which is a synthetic progestin that Linda just talked about that we use. And in these droplets, we gather them up, and we're able to not only culture them, but for a period of 7 days, but do a bunch of different experimental outputs, such as look at time course images, flow sort and sequence the individual monocytes and stromal populations, and also do some downstream analyte detection. So I'll talk primarily about these two first. One of the results that we found is that monocyte proliferation in our co-cultures is blunt in disease. So what we do is that we take a composite image of the stromal cells and CD14 positive monocytes that are encapsulated, and at day 7 of culture, we also do an EDU proliferative stain. And if we isolate just the CD14 positive monocytes, and we can overlay and use cell profiler to quantify the amount of proliferation that the monocytes that we observe in culture. But in disease, it is significantly blunted, right? So that's one metric. But what about other molecular characterizations? So what we did is that we can also dissolve these gels, these co-cultures, and then separate out using flow sort the stromal and monocyte populations. So I'm going to tell you briefly about the monocyte story. So if we were to take volcano plots of the differential gene expression analysis and compare the two different cycle between proliferative and secretory, you can see that in this control group, the monocytes, there isn't a significant difference in the amount of differentially expressed genes. But in disease, there are a lot of genes implicated. And so when you do gene ontology analysis, what you find is that terms related to cytokine production and pro-inflammatory factors such as TNF-alpha or IL-6 are enriched in these gene ontology analysis. So that's interesting. But then the question is, because this is a co-culture model, what are the ligands that the stromal cells potentially are secreting that are pushing monocytes to become dysregulated? So we took a reference data set from our collaborators at the Reagan Institute, Brian Bryson, and they did a very interesting study where they took these CD14 positive monocytes and they stimulated with 60 single conditions, right? So CSF2 or IL-17, IL-7. And what we did is we took our sequenced monocyte data and we plotted it on. So this PCA demonstrates that, okay, the secretory and control co-cultured monocytes don't seem to, at least on this PCA plot, be very different. But in proliferative and menses, you see this kind of drift. Now, this is kind of a dimensionality reduction technique. So I was curious about, can we infer what kind of monocytes these kinds of co-cultured disease monocytes are becoming? So we created gene sets from these reference data set. For example, if I would take differentially expressed genes that are upregulated or downregulated between IL-17 and a control, you would take this and make a gene set, and you can actually run single sample GSEA, gene set enrichment analysis, to compare it to our modeled monocytes. And what this demonstrated was something very interesting, that pro-inflammatory, that we believe that these kind of monocytes that were co-cultured with diseased stromal cells became more pro-inflammatory, okay? So in both kind of the menses and the secretory phase compared to controls. So there is a level of dysregulation there. We also found that in our stromal cell sequencing data, we recapitulated what was observed in vivo, that TYRO3 was actually downregulated, but interestingly, in only the proliferative and the menses model. So there is some kind of cell cycle dependency observed. This is kind of corroborated with what we observed in the monocyte, this kind of drift in both the proliferative and the menses data sets, right? So it points to this dysregulation, but that is cell cycle dependent. And finally, we did a little fancy footwork in terms of trying to understand what are the ligand receptor interactions and how is it disrupted in disease. So we took genes which are from a reference database of cellular communication called OmniPathDB, and we took our data sets For example, we took genes which are stromal ligands and we crossed them with monocyte receptors. And this PCA plot shows that this actually separates in the blue, for example, secretory model separates very fairly distinctly between the Menzies model and also the proliferative model. But in disease, this kind of relationship is significantly disrupted, right? And so the menstrual cycle conditions don't clearly distinguish these ligand receptor pairs. So we're still ongoing analyzing the implications of this. But for example, some pairs which are implicated are prostaglandins or TNF receptors as well. And some of the ongoing work is to understand. We also took the we saved the media, but it's really difficult to distinguish co-culture media. So that's ongoing work as well. But now a little bit more about how we're trying to integrate this into MPS systems. And so in order to recapitulate these cycling hormones, in our model, we actually just had a static addition of estrogen and MPA. However, we really want to move into systems in which we can recapitulate not only the cycling hormones, but control oxygen levels and different additives. To do this, we also, so in that more complex platform, we are currently recapitulating this into a static chip testing platform. So this kind of COC material, which is a plastic, thermoplastic, we also enlarged it so we can do some pre-testing, and I'll show you a little bit of that story right now. So there is a comparable decidual response in this static chip. So if we compare these droplets that I've just talked about to this chip, and we attempt to decidualize stromal cells in it, there is a comparable prolactin secretion over time. And we also demonstrated very recently that we can co-encapsulate stromal cells and monocytes and culture them over a long period of time. So, and in the future, we hope to be able to integrate this kind of stromal monocyte encapsulation with endothelial perfusable networks. And so this kind of is the hope, is that we can potentially push these systems into this kind of multi-compartment integration to understand more to understand the mechanisms behind endometriotic disease. And yeah, this is the overall arching pipeline. And I'd like to thank all the members of the Griffith Lab and of the Laufenberger Lab and all our collaborators. And thank you for your attention. And please go to the posters of Elise Gubbins and Erin Tavonian and Priyatana Roy in the poster session after this. And thank you for your attention.
Yeah, we have time for one question. Here comes Chris.
Hi, thanks very much. I'm interested in these stromal populations that you're putting into a droplet assay. Do they maintain the same phenotype throughout the 7 days, or do they kind of drift or do you keep in the same hormone regimen that they were in at the time they were harvested?
Yeah, so if in the proliferative condition, they were on DMMF12, 5% heat inactivated FBS and estrogen. So but they were first cultured in 2 dimensions. And then in the co-cultures, they are in 3 dimensions. We haven't done, for example, time series RNA-seq to actually understand if in these encapsulations, they do drift, for example. So, but that's a good question for sure. Yeah, but yeah. In the secretory, the plus MPA, they do decidualize, but we haven't done a temporal characterization of only estrogen only. Yeah.